Hello, everybody. Welcome to Ms. McGuire online video lecture. Today, we continue covering human genetics in introduction. And this is part 2 of the lecture, and we will talk about scientific inquiry and scientific method. So please watch the first part of the video lecture to learn about genetics and overview of genetics. Right, so as I said, today we're going to cover scientific method and scientific inquiry. So let's go ahead and begin. Process of science. There are two main scientific approaches. Discovery science is mostly about describing the nature, and hypothesis-driven science mostly about explaining the nature. Right. Discovery and hypothesis-driven. Now, in science, we use two approaches. It's a deduction and induction. Now, you probably heard about deduction and induction many times, and you, I'm sure, know the differences. I just want to remind you that when we're talking about deduction, we go from general to specific. And when we talk about induction, we go opposite, from specific to general. I give you example. For example, I give you example of deductive reasoning, deduction. So you know that birds can fly, right? That's your general knowledge, right? That's kind of applied to all the birds, right? So you know that birds can fly. Now you're looking at some living organism, and this living organism is flying. So your conclusion is it's a bird, right? So you go from general to specific. So you have this knowledge that birds can fly, and now you apply it to very specific living organism. Because it's flying, you deciding it's a bird. It doesn't mean you're right. It might be an insect, for example, butterfly, or it can be even airplane, right? But that's how deduction works. But nothing in science is proven 100%. We always make mistakes. We correct the mistakes, and that's the beauty of science, right? We go from mistake to another mistake to another mistake. So finally, we're getting to the truth, right? Again, so that's deduction. Now, what would be example of induction? Well, induction, you go from specific, from observation to general. So you build up the theory. For example, you study living organism and you study this animal, right? And it can fly. And you're like, oh, it's a bird and it can fly. That's your observations. Then another bird. Oh, this bird can fly too. Oh, here another species of bird. They can fly as well. So that's your, from observation to observation, you come to the conclusion that birds can fly, right? So that's induction. And again, it doesn't necessarily true, because some birds cannot fly. But that give you representation how we use induction and deduction in science. So we start with induction. We do lots of observation, right? So that's your individual, maybe some experiment, another experiment. So you conduct experiment after experiment after experiment. You come to some perception that you're general, right? And then from your general, you use deductive method to come up with a hypothesis about your observations. You use a protocol and analysis, and you come up with a conclusion. And if you support your observation, right, if it's unfalsified, right, so you have your support and support and support. So again, you from individual analysis, right, from individual tests. So you support, and you come up with the theory, right? That's your general. So that's again specific observation, right? Specific support for that particular observation. You come up with the theory, then you use general, that's theory, to make prediction or hypothesis about some specific fact, and you start it all over. right? Or, for example, you do observations. Well, that's a living organism. It can fly. It's a bird. Let's say it's a bird. It can fly. This is another bird. It can fly. Bird can fly. So your perception, bird can fly. Now you use this now general to now you're looking at another living organism and it can fly, right? So you use like, okay, it's a bird, right? Because I already have this perception that bird can fly. You follow protocol, you analyze, and conclusion, let's say it falsifies your previous observation. So then you start over, right? So I know it might be complicated over here. So what do we want to take from this slide? That when we go from general, from theory, from perception to the hypothesis, that's our deductive method. But when we start making observations or doing a test and support our observation, we go from specific, not just specific test, to the general, and then it's inductive reasoning. Oh, right? Lots of fun with this deduction and induction. Now, we just said over here that there is two main scientific approaches. Discovery science is one, and hypothesis-driven science is another one. Now, what is this discovery science? Then when you make observations, it's verifiable observations and measurements that are data of discovery science. So it's not just observation, it's verifiable and measurable. Right? So what does it mean verifiable? So if I make observation about apes and I, keep my journal and I record my observation, somebody can verify them. Somebody can measure them. Right? So observations and measurements are data of discovery science. In biology, discovery science enables us to describe life at its many levels, from ecosystem down to cells and molecules. So, for example, if you take a general biology class and you're using microscopes to make observation of human cells, that's your discovery science. What would be your data? Or you're probably gonna either sketch the cells, draw the cells, label the organelles, or describe them. Or you can measure how big those cells are, how many of them you can see. So that's gonna be your data. Hypothesis-driven science can stimulate us to, this discovery science stimulate us to ask questions and seek explanation, and that arise from your observations. So when you ask questions, you seek explanation, you try to explain nature, then you use hypothesis-driven science. And a hypothesis-driven science used what is called scientific method. That consist of a series of steps that provide a loose guideline for scientific investigation. So here's an example of very simple scientific investigation that use scientific method. So first you start with discovery science, observation. So you discover something. Remote doesn't work. This discovery science bring question, what's wrong? Right? And then actually what is missing over here, you need to do research. So before coming up with a hypothesis, you need to do research about the topic, right? So if you know nothing about remote, that it even has batteries, you cannot come up with hypothesis, right? But you do your research, you learn how remote works, and then you come up with hypothesis, batteries are dead. Then you make your prediction, with new batteries, it will work, and then you experiment. So you just replace the batteries and you try your remote again. And if it works, you support your hypothesis. If it doesn't work, you need to revise your hypothesis. So that's pretty much what it is. Okay. Now, most modern scientific investigations can be described as hypothesis-driven science. Hypothesis is a tentative answer to a question, explanation on trial. Tentative means that it's falsified. You can deny, you can disprove, right? Falsifiable. So it's not always true. Hypotheses cannot be proven true. Make sure you remember this. You can never prove your hypothesis. You can only support or disprove your hypothesis, right? So hypothesis is a tentative answer to a question. It's explanation, it's your prediction. And hypothesis can only be supported or disproved. It can never be proven true, right? So the best hypothesis is in if-then format. If I do this and that, then this would happen due to whatever your reason is. So we will have more examples. Okay, so here's the steps of scientific method, as we already described. So just if you go back over here, that's our steps in scientific method, and now they just in order. So first, observation and research. So we have observation and research. Make observations and research your topic of interest. Problem question. Develop a question or problem that can be solved through experimentation. Formulate hypothesis. Predict a possible answer to the problem or question. Predict the answer to your problem or question. Example. So here's example of hypothesis with if and then. If soil temperature rise, then plant growth will increase. Right? That's our prediction. Next step, experiment. Develop and follow a procedure. Include a detailed material list. Now, why do you need this detailed list? Because again, you always want to make sure that people can repeat your experiment and support it. Next, collect and analyze result, modify the procedure if needed, confirm the result by retesting, include tables, graphs, photographs. Then conclusion, include a statement that accepts or rejects the hypothesis, make recommendations for further studies and possible improvement of the procedure, and communicate the result. So make your result known to other members of scientific community, so they can, first of all, they can support your hypothesis by doing their own experiments, or they can build from your results already. So they don't need to do the same. So they don't need to start with a scratch, right? They can use your data to develop new questions and new experiments to continue your work. Now, parts of the scientific experiment include independent variables that are manipulated variables. It's a factor that is intentionally varied by experimenter. So you're gonna change independent variables. Then we have dependent or responding variables. Is a factor that may change as a result of changes made in independent variables. Now, your dependent variables are your data. So whatever you collect through your experiment, that's going to be your dependent variable. For example, if you go back over here to this experiment that we, to our hypothesis, if soil temperature rises, plant growth will increase. So what are you going to measure? Imagine you conducting this experiment. What are you going to measure? Are you going to measure the temperature of the soil or how tall your plant grow? Right? So what are you going to change over here? Well, for example, you are inside the lab. You're not outside. You're inside a building. Your temperature is constant in the building, right? So you intentionally need to change it. So you're going to change the temperature. and see how this affects that, right? How temperature affect how tall your plant grows. So you're gonna measure how tall your plants are. So that's your dependent variable because this will depend on the temperature, and temperature is independent because temperature doesn't depend on how tall the plant is. So if you plant tall plants, it doesn't change temperature of your soil. It's the temperature of the soil changes how tall plant gonna be, right? That makes sense? So again, so dependent is what you gonna manipulate, what you gonna change through your experiment, and dependent, it's what you're gonna collect, right? It's what will respond to your independent. Now, control group is exposed to the same condition as experimental group, except for the variable being tested. So in the experiment, you always will have experimental group and you will have control group, right? Now, experimental group, you're gonna give them your independent variable. And the control group, you're not gonna give independent variable, right? So for example, and again, if we go back to this hypothesis, if soil temperature rise, plant growth will increase, you will have two groups of plants, right? One gonna be your experimental group. In your experimental group, right, so here's your experimental group. So you will have maybe, you know, like, so you're gonna put here 20 degrees of Celsius. Here you're gonna put 25 degrees of Celsius. So you're gonna change the temperature of the soil and 30 degrees of Celsius. And you're gonna grow plants. And plants gonna grow, for example, short, taller, and really tall, right? So you see how temperature, that is your independent variable, independent, affects your dependent plant's growth, right? So that's your experimental group, but you need your control group. So control group, you would take like, okay, room temperature over here, and you're gonna see how tall your plant's gonna be, right? And let's say your plants are, I don't know, even like this, right? Because room temperature is about maybe 18 degrees of Celsius. So that's gonna be your control group. So in the control group, you're gonna apply all the variables except of your independent, right? So no independent variables over here. Also, you must have either constants. There are several names: constant or controlled or standardized variables. Now, controlled variables are not the same as control group. Controlled variables in experiment are all the factors that the experimenter attempts to keep the same. So let's use our example again with the plant. So here we have our control group, right? So that's control group. And here we have our experimental group. Now remember what we're doing with experimental group. We're changing the temperature, right? So the temperature is our independent. So that's our temperature because we're gonna decide. I put 20, 25, 30, I can put 50, 55, 60, right? I decide. Now dependent gonna be how tall our plants with the increase of temperature, right? In the control group, say like room temperature. Right, so control group doesn't receive this independent variable. Now, what's gonna be a constant in both groups? It needs to be, for example, how much water you use. You need to use the same water everywhere. So you need to water, let's say you use the, I know 1 liter every day. Here also water 1 liter every day. And here also one liter every day. Because if all of a sudden you start using 2 liters over here, then maybe your plants grow taller because of water, not because of the temperature. right? So everything you need to keep constant except of independent variables, all right? So that's, those are important part of scientific experiment. Now, also when you graph your data, your independent variables on the x-axis and your dependent variables on the y-axis. So here's another example. So here you have two identical pods, right? Two identical pods and 10 identical bean seeds. Now, one gonna be experimental group and one control group, right? So that's your experimental and control group. Now, you wanna research how watering plants affect germination of plants. How many of these seeds will sprout? Are they sprout because of water, or they just gonna sprout without water? Who knows, right? Maybe you don't need to use water and they're gonna sprout anyway. So you're trying, your hypothesis here, if I water bean seeds, they will sprout, right? If I water, they will sprout. Now, if I don't water, they will not sprout. And you start with, again, that's your experimental group, that's your control group. Control group doesn't receive your independent variable. Your independent is water, right? Control doesn't receive it. So you water, and then your results, 9 of 10 sprout, 0 of 10 sprout. So you can support your hypothesis that yes, if you water your bean seeds, they will sprout. Your constant over here are identical pods, same number of seeds, and same type of seeds, bean seeds. You're not gonna put here, I don't know, some sunflower seeds, and here you put your bean seeds. So you keep everything constant except of independent variables, that is water. Now your independent variable here is water. Your dependent variable here is how many seeds actually sprout. And your controlled variables here, identical pod, number of seeds, type of seeds, that's your controlled variables. Your control group is the one that does not receive independent variable, and your experimental group, the one that receives independent variable. Oh, okay. So another one, proper experimental design. Objectivity of science, and especially medical science, depends on property designed experiments. So experiment must include adequate sample size, control group, and experimental or treatment group, prevention of psychosomatic effects, that kind of like being biased, because if you conduct your experiment, you want it to be successful, right? So, and maybe people in your group, if it's not animals, because we're talking about medicine here. So for example, you work with people and they want you to be successful as well. So that's why you use placebo, right? So for example, you develop a drug and you want to see how this drug affects blood pressure. So you have your control group, 10 people, and experimental group, 10 people. However, I don't think it's adequate sample size, but maybe 100 people here. But let's even, well, sometimes you can start with a small sample size. Now, this 10 people who are your control group, you're going to give them placebo. They have no clue. They're thinking you giving them medication, but you don't. But that's how you prevent this psychosomatic effect. And your experimental group, you're going to give them your medication. And then you see the results. You collect results, how their blood pressure, right, with placebo and with your medication. Also, we need to eliminate experimenter bias. we use double-blind method. And that means not only people do not know if they're getting placebo or not, but you yourself, who is experimenter, you also don't know who gets placebo and who gets your drug. This way, you're not biased. Then also statistical analysis of the significance of the data and peer review by other experts. Right, so here's our double-blind experiment that we just discussed when we have patients and neither one of them knows who getting a drug and who getting the placebo, and a doctor itself doesn't know who gets what, so he or she cannot be biased either. Right. Okay. Culture of science. Now, scientific theories are not the only way of knowing nature. Science, religion, and art are very different ways of trying to make sense of nature. However, science is different. Science has two key features that distinguish it from other forms of inquiry. Science depends on observation and measurements that can be verified and requires ideas or hypotheses are testable by experiments that others can repeat. Right, so if you in a religion or in art, or even you in the philosophy field, neither one of them, art, religion, or philosophy, doesn't require these ideas to be verifiable and testable, but science does. So it must to be testable and it must to be verifiable. Okay, so here's very important, culture of science. Science uses scientific method that we just described. Remember, scientific methods have special steps: observation, research, hypothesis, experiment, collecting the data, analyzing the data, making conclusion, then include precise procedure, and then share your findings. That's all scientific method. You must have dependent, independent variables, control group, experimental group, constant variables. So that's all what we discuss at scientific method. Another important culture of science, that ideas must be tested. So you cannot just come up with the, let's say, hypothesis and explanation without experiment. You must have experiment. Another, science can only look at natural world and no absolutes. Scientific laws are not considered absolute truths. In fact, the core philosophy of the scientific method is that no scientific fact is to be considered an absolute truth. You see, even scientific fact, and when we hear fact, that must be true. Well, no, even a fact in science is not absolute truth. And by the way, we're gonna look what fact is in a minute, right? So now, what is a natural world? A natural world is anything that is made of matter, and matter made of elements. And elements you will find in periodic table of elements. So if it's made of elements that are in periodic table, right, or maybe some newer elements that we will discover in future, so the matter that has mass and occupies space, then it's going to be natural world, right? So everything is, let's say your desk is part of natural world because it's made of wood, and wood is made of cellulose, and cellulose is made of carbon and hydrogen and oxygen, right? So it's a part of natural world. You are part of natural world. Now, what is not then a part of natural world? That Okay, so let's just kind of, I'm ahead of myself. So in the language of science, the term natural refers to any element of the physical universe. What's natural is often identified as testability. Natural things behave in predictable ways, though we might not yet fully understand them, which have observable outcomes. The predictability means that we can test hypotheses about natural entities by making observations. Ghosts, for example, are supernatural entities without a basis in the physical universe and so are not subject to the laws of that universe. Hence, ghosts are outside the pure view of science, and we cannot study their existence or lack of them with the tools of science. All right, so let's go back. So that tells us that we look at natural world only. And again, remember, no absolutes, even facts in science cannot be absolute truths. And this is fundamental core of science. It's so important to understand, because if you ever hear some scientist or whoever names a scientist make an argument that that's true, that's absolute true because I know it or I proved it, then you know that's not how you practice science. Okay, but now let's look what fact is. Fact is observation that has been repeatedly confirmed and accepted as true. But look, true is in parentheses. Truth in science, however, is never final. Now, hypothesis is a tentative statement about the natural world. It must be testable. Experiment is a support or disprove a hypothesis. Law, a descriptive generalization about how a part of the natural world behaves under stated circumstances. Laws are the basics for theories, and laws may or not be facts. Theories are a well-supported explanation of some aspect of the natural world that can incorporate facts, laws, and inferences and tested hypotheses. So now look, this is the most important probably slide to understand what theory and fact and hypothesis is in science. This is what most people assume. That is not true. People assume that, well, hypothesis is something that we need to test. We don't know if it's true or not, right? From hypothesis, we go to the theories, but above the theories, we have laws, and then we have facts. That's not how it is in science. In science, actually, we're going to start with facts. That's our observation. And we make this observation that, you know, it's they became acceptable as a fact, but it can only exist on our planet. So, for example, what can be a fact? The fact is that living organism is made of carbon. We have carbon-based life. That's a fact, because you make like hundreds of thousands observations. You study any living organism over here, and you will find that they are based on carbon. That's a fact. But then you go to a different planet and life is not made of carbon over there. It's completely wrong. They may be made of silicon, right? So that's a fact only based on what we see here on our planet, right? Then you make a hypothesis from the fact. From the hypothesis, you through the experiment, you come up with a law, right? So law, they state the phenomena, right? This is how it works. But theories explain how law works. So now let's go back. So just remember that we start with the observation or fact. We use them to make a hypothesis. When hypothesis is supported over and over, we have a law, and the theories are even above the law. Now look over here, we compare law and a theory. So a law is just summarize a set of observation about natural phenomena. And theory explain why natural phenomena occurs. But they both have, of course, similarities. They both based on hypotheses that can be used to make prediction and can be revised. Right? So nothing, you can revise a law and you can revise a theory. Right? So we say law, like law of gravitation. We know we have gravitation again on our planet, but it's only hold truth again on our planet only. If we in an open space, then we don't have gravitation. All right, but theory explains how this gravitation works. Right, so and it's important to understand because especially when we talk about theory of evolution, for example, and people say it's just a theory. Because theory sounds like, okay, well, somebody just decided all of a sudden that this is how things work, and it's just nothing but a theory. But remember, theory is actually based on facts, hypotheses, and laws. It explains the law. It's supported by hypothesis, and hypothesis was driven from a fact that we make observation of these facts over and over, and it's retestable, it's a verifiable observation. Okay, I think that's our last slide. Thank you for watching. Don't forget to watch the first part of the lecture. If you have any questions, let me know. And I hope it was helpful. I talk to you next time. Bye.